Okay. In the previous video we have discussed about what? The factors affecting crossing over, and let us discuss the next part of the chapter, that is arrangement of linked genes on a chromosome. Okay. In the previous video we have completed the factors affecting crossing over. And what are the factors affecting crossing over? Distance, temperature, X-rays, age and the sex. These are the 5 different factors that affect crossing over. Okay, these are the 5 different factors that affect the crossing over. What happens in case of distance? If it is distance is more, then crossing over will be more. Like the same, if the temperature is more, crossing over will be more. And X-ray, if it is more, then crossing over will be more again. If age is more, then crossing over will be less. If the individual is male, then the crossing over will be less again. About this we had discussed in our previous video and we have finished it. Coming to the next part of the chapter, that is arrangement of linked genes on the chromosome. Okay, arrangement of linked gene on the chromosome. On the particular chromosome, how exactly the genes are arranged. Okay, there are 2 forms in the arrangement, that is cis arrangement and trans arrangement. Okay, cis arrangement and trans arrangement, 2 different types of arrangements of the genes are there. Particularly arrangement of the linked genes are of 2 types, that is cis and trans. Coming to the first one, that is cis arrangement. What is cis arrangement? See, when 2 dominant genes are located on one chromosome and both recessive genes are located on another chromosome. Okay. See, if you consider this as one chromosome and this as another chromosome, that a chromosome number A and chromosome number B, on this chromosome number A, 2 genes will be present and both the genes will be dominant. Okay. If both the dominant genes are present on one chromosome and if both the dominant genes are both the recessive genes are present on another chromosome, these are recessive. Okay. If one chromosome contain both the dominant genes and another chromosome contain both the recessive genes, then that type of arrangement of the genes is termed as cis arrangement. Okay, such type of arrangement is termed as what? Cis arrangement. Okay. Next, do remember that the important point is regularly, that means naturally, cis arrangement will be seen. That means the original point or the original arrangement of the genes will be always in the form of what? Cis type or cis arrangement. But when you see the next one, see, that is a trans arrangement, just exactly opposite to that of the cis arrangement. See what happens in the trans arrangement, a chromosome bear one dominant and one recessive gene and another chromosome also possess one dominant and one recessive gene. Okay. See, in case of trans arrangement, what will happen? 2 chromosomes will be there, that is A and B, but on these one dominant will be there. Okay, one dominant gene will be there and another one will be recessive. Okay. So, this type of arrangement is called as what? The trans arrangement. In case of cis arrangement, see, the genes on one particular chromosome both will be dominant and another particular chromosome will have both the genes are recessive. But in case of trans arrangement, what will happen? One chromosome will have dominant and recessive, even the other chromosome will have dominant and recessive gene. Is that clear? This type of arrangement is called as what? Trans arrangement. And trans arrangement is not an original form, that means it is occurred due to crossing over. Very important point here to be noted. Okay. Trans arrangement is occurring due to what? Crossing over. And 2 types of gametes are also formed in the trans arrangement. Okay. But it is different from cis arrangement. Right. So, next one, sex determination, coming to the most important point of the chapter and very important point for your even theory examination, which will be asked for 5 marks, right. which will be asked for 5 marks in the theory examination, but in case of NEET examination, even the CET examination, about the sex determination, you may hardly expect, or you may expect strongly one question on this one. Okay, one question can be strongly expected from sex determination, right? What is sex determination? Coming to the question, what is sex determination? See, it is establishment of sex through differential development. Establishment of sex through differential development in an individual at an early stage of developmental life is called sex determination. It means when an organism is to be produced, it should be given with a particular sex, whether it should develop into male or it should develop into female, and the determination of or the fixing of the sex of the individual, whether it should develop into male or female. So, that particular phenomenon is termed as sex determination. ಅಂದರೆ ಒಂದು ಯಾವುದಾದ್ರೂ ಜೀವಿ, ಓಕೆ, ಒಂದು ಯಾವುದಾದ್ರೂ ಆರ್ಗ್ಯಾನಿಸಮ್ ಏನಾದರೂ ಹುಟ್ಕೊಂತು ಅಂತಂದರೆ, that organism whether it should develop into male or it should develop into female. Okay, so that particular part which is deciding the sex of the individual, ಅದನ್ನೇ ಏನಂತ ಕರಿತೀವಿ, sex determination ಅಂತ ಕರಿತೀವಿ. That is deciding sex of an individual, whether it should develop into male or female. So, that itself is called as sex determination. Okay, it is called as what? Sex determination. Next one, various methods operate in the sex determination. That means in the sex determination, okay, to determine a particular individual's sex, there are different varieties or there are different methods. Okay, they are, first one, environmental sex determination, second one, non-allosomic genetic determination, third one, allosomic sex determination, and haplodiploidy sex determination. Okay, different types of sex determinations are seen. Okay, let us go through one by one. Okay, let us go through the different types of sex determinations one by one, right? See, in the first one, sex determination based upon fertilizations are of 3 types. Okay, sex determination based on fertilizations are of 3 types. Which are they? The first one, progamic, okay, progamic sex determination, second one, Singamic, third one, epigamic. What is progamic? Okay, progamic sex determination, okay, it is nothing but the sex determined before the fertilization. Okay, the sex is determined before the fertilization, that is called progamic. Before the fertilization, if the sex is determined, it is called as progamic, right? Example, the drone in the honeybees. Okay, the drones of the honeybees are what? Progamic in nature. That means drone is the male individuals, and these male individuals sex is determined before the fertilization. Okay, if fertilization occurs, then the fertilized egg will give rise to what? The female individual. Okay, the fertilized egg give rise to female individual. So, without fertilization, the sex of the individual, if it is determined, it will be what? Before the fertilization. So, that is honeybee drone. Next one, Singamic sex determination. That means sex determination occurs during what? Fertilization. Okay, if the sex determination occurs before fertilization, then it is called progamic. If it occurs during fertilization, then it is called Singamic. What do you mean by Singamy? Okay, what do you mean by Singamy? It is the process of, Singamy is nothing but what? The process of fusion of the male and female gamete. Yes or no? Male and female gametes. The process of fusion of male and female gametes is called as Singamy. Is that clear? So, Singamic type of sex determination is the sex determination in which sex of the individual will be determined or fixed during what? Fertilization. Example, most of the plants and animals. Next one, epigamic epigamic sex determination. Epi means after, okay, epigamic sex determination, it is determination of sex after the fertilization. Very important point here. The sex of the individual will be determined after the fertilization. That means after the fusion of male and female gamete, right? Example, females in case of honeybees, okay, females in case of honeybees. How exactly the sex is determined in the female honeybees? Very important, okay. The honeybees, see, I will explain here the sex determination, okay, very clearly about the honeybees. Honeybees will lay the eggs. If the egg is developed without fertilization, okay, without fertilization, it will develop into male. Do remember, okay. If the egg is developed with fertilization, it will develop into female, but what happens? It will be sterile in nature, okay. The fertilized egg will develop into what? A female, but that female will be sterile in nature. That is why we call the sterile females as workers, okay, it is called as worker. And if the egg after fertilization, okay, to be developed into a female, okay, to be developed into a female, okay, active female, that means fertile female, that particular baby or else that particular individual should be fed with a material called as royal jelly, okay. If the larva is fed with royal jelly, then that particular bee will develop into a female, fertile female individual, okay. In case of honeybee, to give rise to a fertile female individual, what will happen? Royal jelly will be fed to the fertilized, okay, eggs. Fertilized egg which will give rise to a particular larva, to that larva if royal jelly is fed, then it will develop into female individual. So that type of sex determination is seen and it is called as epigamic, which will be taking place after the fertilization. That type of sex determination will take place what? After the fertilization, right? Next one, mechanism of sex determination. See, mechanism of sex determination. Wilson and Stevens proposed chromosomal theory for sex determination. Very important point. Who have proposed chromosomal theory of sex determination? Okay, that is Wilson and Stevens. What they have done? Okay, they told that chromosomes are of two types. They are autosomes and allosomes. And do remember, autosomes are also called as somatic chromosomes. They regulate somatic characters of our body. Autosomes or the somatic chromosomes, they regulate somatic characters of our body. What do you mean by somatic characters? Somatic characters are nothing but non-sex characters, okay, non-sex characters. That means the regular wear and tear growth of our body, the bones, the body organ systems, okay, they are all what? The body characters, and which do help for only the mechanism to develop in the body, okay, the development of the body, right, and they will not take part in the reproduction. The parts which do not take place in the reproduction or which do not take part in the reproduction, they are called as somatic characters or somatic parts of our body. And the autosomes, okay, autosomes or somatic chromosomes will be leading to or will be managing the somatic parts and somatic characters of our body. And allosomes are there, which are called as heterosomes or sex chromosomes. These are the chromosomes which are not at all concerned with our body growth and any of the part of our body except the sexual development. Okay, the sex chromosomes always help for what? Determining the sex of the individual. Okay, they are associated with what? Determining sex of the individual, whether the individual should develop into male or female. So that is called as what? The sex determination with the help of chromosomes, which was proposed by Wilson and Stevens. Okay. Next point, see here, sex determination was first discovered by McClung in grasshopper. Okay, sex determination was first discovered by McClung in the grasshopper. X chromosome called as X body, okay, because when it was observed under what, the microscope, it was looking something like a structure, dark body, and they were not knowing what exactly the material is. That's why they have given the name X. Okay, let us consider it as X. In that way they have given, let us consider this as X body, and now today we are going to call that X body as X chromosome. Okay, we are calling that X body as X chromosome. Okay, X chromosome or X body was discovered by Henking. Okay, when he found that in the testis of male bug, okay, in the testis of male bug, one chromosome has no homologue. Okay, in the male bug, okay, in the male bug, okay, that too, when Henking was observing in the male bug, he found that one chromosome was there without any homologue. That means to that particular chromosome no homologous chromosome was present again, and he called it as X body, and nowadays we are going to call that X body as X chromosome, right. Next one, there are 5 types of sex determination mechanisms. Coming to the next part of the chapter, that is mechanisms of sex determination, right. In the mechanism of sex determination there are 5 types, okay, 5 types of sex determination mechanisms are there. First one, see, XX and XY type of sex determination. So what do you mean by XX and XY here? These are the sex chromosomes, okay, XX and XY are the sex chromosomes, right. So first type of sex determination is XX and XY. It is a type of sex determination in which female is homogametic. That means the female will have same type of gametes. It means if autosomes are there, along with the autosomes, okay, female will have X and X. Okay, when she produces the gamete, she will give rise to A and X and A and X. Both the gametes will be same. That is why it is called as homogametes. Okay, the female will be homogametic in nature. And what about the male? Male will have autosomes along with X and Y. Okay, they will produce what? Autosome plus X and autosome plus Y. So these two are different here, AX and AY. That is why the males are called as heterogametic. Okay, males are called as heterogametic, but the female is called as homogametic. In XX and XY, it is a type of sex determination in which female is homogametic, that is produces only one type of gamete, that is autosome along with the X, right. And in male, X chromosome containing gamete is called as gymnosperm and Y chromosome containing gamete is called as androsperm. Okay, that means the males can produce autosome along with X and autosome along with Y, and the sperm or gamete having autosome along with X, it is called as what? Gymnosperm. It is called as what? Gymnosperm. Okay, male gamete is called as sperm, and male gamete with X is called as gymnosperm. The same male gamete with Y chromosome is called as androsperm. Okay, it is called as what? Androsperm, right. Male gamete with X chromosome is called as gymnosperm and Y chromosome is called as androsperm. Examples in case of human beings or in case of man, in case of Drosophila and dioecious plants like the coccinea and the melandrium, you can see this particular type of XX and XY type of sex determination, okay, where the male individuals will have autosomes along with X and Y, the females will have, this is what? Male, okay, and the females will have autosomes along with X and X. This is female. Clear? So, this is XX and XY type of sex determination. Next comes the another one, that is ZZ and ZW type of sex determination. Okay. It is another type of sex determination wherein which the female will be, okay, I will first make it clear here. Okay. See, it is another type of sex determination in which female will be what? Heterogametic again. Okay, as it is XX and XY, here we are going to mention ZZ and ZW. Okay, like the XX and XY where female was homogametic, here the female is heterogametic. Unlike the XX and XY, it is different. Okay, in the XX and XY, female was homogametic. Okay, but here in ZZ and ZW, female is heterogametic. That means along with the autosomes, if ZZ and ZZ is present, along with the autosomes, if it is Z and Z is present, it will give rise to male. Okay, if along with the autosomes Z and W is present, it will give rise to the female. Okay, since Z and W are the two different sex chromosomes present in the female here, okay, it will produce two gametes with autosomes along with Z, like autosomes along with Z and autosomes along with W. ಅಂದರೆ ಇಲ್ಲಿ ಎರಡು ತರ ಗ್ಯಾಮೆಟ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತವು ಫೀಮೇಲ್ಸ್ ಅಲ್ಲಿ. ಒಂದು ಅಲಾಂಗ್ ವಿತ್ ದ ಅಟೋಸೋಮ್ Z ಇರುತ್ತೆ, ಇನ್ನೊಂದು ಅಲಾಂಗ್ ವಿತ್ ದ ಅಟೋಸೋಮ್ W ಇರುತ್ತೆ. ಓಕೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ವಾಟ್? Okay, that is called as the ZZ and ZW type of sex determination, and in this the female can produce two different types of gametes which is called as heterogametic. Okay, the female heterogametic. Okay, the female heterogametic it is called as. Next one, but in these the males individual or male individuals will be homogametic, opposite to that of XX and XY. Okay, opposite to that of what? XX and XY. Okay, next one, it is found in some insects like butterflies, moths, vertebrates like birds, fishes and reptiles. Okay, commonly, most commonly it will be found in the butterflies, moths and vertebrates, and in higher organisms such as birds, fishes and reptiles it can be found. Next point, in case of plant kingdom, this type of sex determination is found in the what? See, Fragaria elatior. Okay, Fragaria elatior, right? So, these are the two important types of sex determination. Coming to the next type, okay, coming to the next type, that is the XX and XO type of sex determination. XX and XO type of sex determination. Okay, coming to the next one. Okay, let us continue with the same thing. Okay, that is XXXO type of sex determination. The next one is XX and XO type of sex determination. So, this is the type of sex determination in which, okay, the females will have, okay, the females will have, see here, the female is homogametic. That means the female will have, along with the autosomes, they will have X and X. The female will have two X chromosomes along with the autosomes. That is why they can produce homogametes. That means autosome along with one X chromosome and autosome along with one X chromosome. These will be the gametes which are produced by the females. That is why the females are homogametic. But male is heterogametic. Okay, male is heterogametic. What happens in the male? Male will have autosomes, along with the autosomes only one X chromosome will be there, and in another sex chromosome there will be nothing. Okay, there will be nothing, which is represented by the letter O. Okay, 0 it means. That is why what will happen? Two types of gametes are produced. One gamete will be along with the autosome X, and another gamete will be along with the autosome there will be nothing. We can represent it as O. two different types of gametes will be produced here. That is why in XX and XO type of sex determination, the females will be homogametic and males will be heterogametic. And this type of sex determination is found in the grasshopper as well as the squash bugs, that is Anasa, as well as in the cockroach, okay, in the ascaris, and in the plants like the Dioscorea sinuta and Vallisneria spiralis. So, all these are the examples for what? The organisms which show XX and XO type of sex determination. Okay, these are the organisms which show XX and XO type of sex determination. Is that clear? Where the females can produce similar type of gamete, that is AX and AX, but the males can produce dissimilar gamete, that is AX and AO. Very important point to be remembered here. What do you mean by this O? It means nothing or it means 0. Okay, it means nothing. Okay, it means nothing or 0. Okay, there will be no chromosome, okay, along with X in the males, right, which can be represented by O there. Next point, okay, next fourth type of sex determination, that is ZO and ZZ type of sex determination. Okay, it is a type of sex determination in which female had odd sex chromosome while the males have two homomorphic sex chromosomes. Okay, that means in case of males, along with the autosomes, Z and Z will be there, but in case of females, along with the autosomes, only one Z will be there. So that itself is called as what? Okay, the female had one odd sex chromosome. Okay, odd sex chromosome means it has got only one sex chromosome. Okay, but along with this Z, there will be nothing in front. Right, this type of sex determination is called as ZZ and ZO type of sex determination. And here males will produce, okay, the males will produce homogametes, that means only one type of gamete or similar gamete, but females will produce what? Two different types of gametes. Okay, females will produce two different types of gamete. Is that clear? And in case of cockroaches and grasshoppers, okay, it will be observed exactly opposite. Right. Next one, Haplodiploidy. Okay, Haplodiploidy type of sex determination, which is the type of sex determination found in what? Honeybees. Okay, honeybees shows haplodiploidy. That means what happens to the particular egg which is young or haploid in nature, okay, egg. If this egg develops without fertilization, okay, without fertilization, then what happens? It will develop into male. Without fertilization, if it develops, it will develop into male. If the same egg, okay, develops without fertilization, that means it will be in the form of haploid condition only. It means males will be haploid in nature. Okay, but if the same egg is fertilized by a sperm, it will become what? Diploid. And if it gets fertilized by a sperm, when it gets diploid, then it will develop into the female. Okay, and this female individual, if it is fed with, okay, if it is fed with royal jelly, if it is fed with royal jelly, it will become fertile female. But if it is not fed with royal jelly, then it will become sterile. Okay, it will become sterile. But even though it is sterile, it will be what? Female itself. Okay, even though it is sterile, it will be female itself. That is why in case of honeybees, what type of sex determination take place? Haplodiploidy. It means if the haploid egg cell directly develops into an individual, it will give rise to male. Okay, if the egg cell is fertilized, it forms diploid condition, and the diploid condition, if it is formed, then the individual developed will be what? Female. Okay. The haploid individuals will be male and diploid individuals will be female, and it occurs in honeybees and also in case of wasps and ants. Okay. In honeybee, see here, male individual drone develops from unfertilized eggs, and male is always, okay, the parthenote. Okay, male is always the parthenote. Queen and worker bees develop from diploid eggs, that is fertilized egg. Okay, queen and the workers are developed by diploid eggs, or it is also called as fertilized eggs. So, this type of sex determination is called as haplodiploidy. Okay, haplodiploidy, right. Next one, coming to the most important point, that is about the human beings. Okay, sex determination in human beings. As we have already studied in the previous slide, that is, in case of human beings, sex determination takes place with the help of sex chromosomes, that is X and Y. If along with the autosomes, okay, if along with the autosomes X and X are present, then it will be leading to what? The female individual, okay, female, and the male will be produced by means of what? Autosomes with X and Y chromosome. Okay. Along with the autosomes, if X and Y chromosome is there, it will lead to male, and along with the along with the autosomes, if X and X chromosomes are there, it will lead to female. So, the same thing is explained here very importantly. See here, human beings have 22 pairs of autosomes, it means 44 autosome chromosomes. Is that clear? 22 pairs of autosomes means a total of 44 autosomes. And what are 44 autosomes? They are nothing but 44 chromosomes which are responsible for the maintenance of body characters, not the sex characters. But along with this, the sex chromosomes will be occurring, which will make the individual to develop into male or female. Okay. See here, there occurs a special gene on differential region of Y chromosome. Okay. That is called as Sry gene. Okay, that is called as Sry gene. Okay. Particularly, this Sry gene will be present in sex determined region on the what? Y chromosome. It will be present on Y chromosome, right. This Sry gene forms a proteinaceous factor called as TDF, nothing but what? Testis determining factor. Okay. By the time, I would like to give you one information here, as we have already discussed in the previous class of the same chapter. Okay. That is, TDF is the smallest gene. Okay. TDF is the smallest gene, right. The testis determining factor. Okay. And this testis determining factor is responsible for what? It is responsible for development of male reproductive organs. Okay. So, presence and absence of Y chromosome determines the sex in the human beings. If along with the 44 autosomes, if the individual has got X and X, okay, in absence of Y, I mean, okay, it will develop into female. Why? Because since the Y chromosome is absent, the Sry gene will be absent. If Sry gene is absent, then TDF cannot be produced. What is TDF? Testis determining factor. It cannot be produced. That is why the individual will develop into female. But in case if along with the 44 chromosomes, okay, autosomes, if X and Y is present as the sex chromosome, then what will happen? It will develop into male individual. Why? Because presence of Y chromosome is there. And what the Y chromosome contain? Y chromosome contain Sry gene. Okay. What this Sry gene have got, or what it can produce? It will produce a TDF, testis determining factor. Yes or no? by means of testis determining factor, the male reproductive organs are developed. Is that clear? By means of testis determining factor, what will be developed? Male reproductive organs can be developed. Due to this, the individual which have got 44 autosomes along with X and Y chromosome will become or will develop into male individuals, and the individuals which have got 44 autosomes along with X and X sex chromosomes, they will develop into female individuals. So, here the point is very clear that if the individual have got Y chromosome, it will develop into male. If the Y chromosome is absent, then the individual will develop into female. Is that clear? If Y chromosome is present, they will develop into male, and if Y chromosome is absent, then they develop into female. Is that clear? And very important point, let us see whether it is mentioned. No. Okay, very important point here. Okay, very important point here. Okay, I would like to write it very clearly. See, in case of human males, okay, in case of human males, they have got 44 X and Y. Okay, the genetic content of human males is 44 X and Y, but human females have got 44 X and X. That is why the male individuals can produce two different types of gametes, which is called as male heterogamity. Okay, they can produce 22X and 22Y, two different types of gamete. So, this condition is called as male heterogamity. It is called as male heterogamety, right? And females will produce only one type of gamete, that is 22X and 22X, right. So, that is why the females are called as what, or this condition in which the female can produce only one type of gamete is called as female homogamety. Okay, that is called as female homogamety. Clear? Male heterogamety and female homogamety, right. By this one point I would like to make you clear, the males will produce two types of gamete, that is 22X and 22Y, and females will be producing only one type of gamete, that is 22X. And see here, if you consider this as egg, one egg, and this is another egg, right. If the sperm having 22X fuses with the egg, it will lead to a formation of 22X and 22X, which will lead to formation of what, 44X and X, and this will develop into female because there will be no presence of Y chromosome. And at the same time, if another sperm having Y chromosome fuses with the egg, okay, Y chromosome, the another sperm having Y chromosome fuses with the egg, it will lead to what? The formation of 22X and 22Y, it will lead to 44X and Y, which will develop into male because of presence of Y chromosome. And see here, the genes of Y chromosomes are known as Hollandric genes. Okay, the genes of Y chromosome are known as what? Hollandric genes. I will write it here, Hollandric. The genes of Y chromosome or male or masculine genes are called as Hollandric genes, male genes. Clear? Next one. Okay. Coming to the next part of the chapter, that is mutation. Okay. Next part of the chapter is mutation. What is mutation? The first point is, what is mutation? So, here I would like to explain what is mutation first. Mutation is nothing but, see, mutation is nothing but sudden inheritable change in the genetic material. Okay, very clearly it is mentioned. Okay, genetic material, it may undergo changes sometimes, but if it is not inheritable, then it cannot be called as mutation. Okay. If it can be given to the next cell or next generation, then it can be called as what? Mutation. Okay. Sudden inheritable change in the genetic content of an organism is called as mutation, right? And mutations are discontinuous sources of variation. See here, they are not continuous sources, but they are discontinuous sources of variations. The regular variations are occurring due to what? Crossing over, but the mutation may also lead to variations. That is why it is called as discontinuous source, right? And the term mutation was coined by Hugo de Vries. The term mutation was coined by the scientist called Hugo de Vries. And Hugo de Vries studied mutation in the plant, that is Enothera lamarckiana, okay, that is evening primrose. Enothera lamarckiana, it is the scientific name of the plant, and the common name is evening primrose. Okay, evening primrose. The evening primrose he had studied properly, and in that particular plant de Vries observed what? The mutation for the first time, right? Next, he observed 834 mutations in 54,343 plants of Enothera lamarckiana. See, in how many plants he have observed? Is a very huge number of plants he have studied, that is 54,343 plants. In these plants he has observed almost 834 mutations. Okay, the credit of discovery of mutations is given to Morgan. Okay, the credit of discovery of mutation is given to Morgan. He observed some white-eyed male Drosophila in a population of red-eyed Drosophila. Is that clear? Both of them have observed the mutations, but the credit of mutation is given to Morgan, Th. Morgan, right? In a Drosophila, eye color is a sex-linked character, as you already know. In Drosophila, eye color is sex-linked character. That means the gene for eye color in the Drosophila is present on the sex chromosome. Hence, it is called as sex-linked character. And the gene of eye color is located on X chromosome. The gene of red eye color is dominant over the gene of white eye color in the Drosophila. See here, in Drosophila, eye color is sex-linked characters. Okay, and on which particular chromosome it is present? On the sex chromosome, that is X. The gene for eye coloration in case of Drosophila is located on X chromosome. Okay, it is located on what? X chromosome. Clear? And it has got two variants or two alleles, that is the red eye and white eye, where the red eye is dominant over the white eye. Right. Next one, chromosomal aberrations. Next, coming to the next part of the chapter, that is chromosomal aberration. What do you mean by aberration? Aberration means what? Change in the structure. Okay, change in the structure, that is chromosomal aberration. Aberration means what? Change in the structure. Okay, or abnormalities, you can also call it as what? Abnormality. Okay, chromosomal aberrations means chromosomal abnormalities or the defective structures of the chromosome. Okay, what is chromosomal aberration? See here, it is change in the structure of chromosomes, and it is of four types. First one is deletion, second one inversion, third one duplication, and fourth one is translocation. Okay, let us go one by one. Okay, the first chromosomal aberration is deletion. Okay, or the first chromosomal aberration or the structural loss is what? Deletion or abnormality. Chromosomal abnormality, in that the first one is deletion. What is deletion? See here, it is the loss of a part or a segment of the chromosome which leads to loss of some genes. See here, if you consider this as a chromosome, okay, and if a part of that is deleted, then what will happen? the genes in that particular part of the chromosome will be lost. Yes or no? So, that itself is called as what? The type of chromosomal aberration which is deletion. You can see, say for example, in case of Cry-Du-Chat syndrome, okay, Cry-Du-Chat syndrome, in that what happens? The chromosomal part of 5th chromosome, a part of 5th chromosome will be lost. So, that is called as what? Cry-Du-Chat syndrome, okay, where the 5th chromosomal part will be lost, okay. Due to that, some of the genes will be lost also. Second one, that is inversion. See here, invert means, okay, invert means to change, okay, in a reverse manner, to change in a reverse manner. See here, inversion is breakage of chromosomal segment but reunion on the same chromosome in reverse order. So, that is called as inversion. Say for example, if a segment of DNA is there like this, okay, if a segment of DNA, on these some chromosomes are there, A, okay, B, then C, then D, okay, E, then F, G. So, these are the different chromosomes, or these are the different, what you can call it as genes are there in this particular segment of the DNA, okay. What happens in the inversion is breakage of chromosomal segment will be taking place. That means a part of it will be broken down, it will be separated, broken down and separated. But what happens after separation? Reunion will take place, but it will not reunite in the same proper way, but it will be undergoing reunion in the reverse order, okay. What will happen? It will lead to formation of a strand like this. See here, A, then B, as it is, no problem will be there, but CD will be reverted, or it will be reunited in the reverse manner. That means D comes first, then comes the C. So, because it is inverted and shifted here, okay, then the further things E, then F, then G will remain as it is normally. So, this process is called as inversion, okay, it is called as what? Inversion. All the genes will be there as it is, but they will be in altered form, or there will be in what? Altered position, right. Next one, duplication. What is duplication? Very simple, by the name itself you can understand, it will be duplicated. That means a part of the chromosome will be duplicated or copied, or it will be occurring twice, okay. See, what is duplication? The occurrence of a chromosomal segment twice on a chromosome, okay, occurrence of chromosomal segment twice. See here, I will write, if the A, B, then C, D, C, D, E, F, G. So, here what has happened? C and D, C and D, they have occurred twice. Yes or no? This itself is called as what? Duplication. And the condition is here, okay, the point to be noted is the C and D, or part whichever is duplicated, may not be, or it may not be present in the same next part, or it may be present in the other part of the same chromosome also. Andre iದೇ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಮೇಲ್ಗಡೆನೇ ಇರುತ್ತೆ, ಆದರೆ ಇಮ್ಮಿಡಿಯೇಟ್ಲಿ ನೆಕ್ಸ್ಟ್ ಇರಲಾರ್ದೆ ನೆಕ್ಸ್ಟ್ ಬೇರೆ ರೀಜನ್ ಅಲ್ಲೂ ಡೂಪ್ಲಿಕೇಟ್ ಆಗಿರಬಹುದು. ಓಕೆ, ಇಟ್ ಮೇ ಗೆಟ್ ಡೂಪ್ಲಿಕೇಟೆಡ್ ಓಕೆ in other regions of same chromosome, okay, not always in the next position. Clear? Next one, the next part or next type of chromosomal aberration is translocation. Okay, by the name itself you can understand translocation. It means the location will be changed completely. See here, a part of chromosome is broken and may be joined with non-homologous chromosome. It means if you consider this as the part of the chromosome, okay, and this is another chromosome, okay, here A, B, then C, D, E, F and G, and here it is another non-homologous chromosome which will be having, okay, O, P, Q, then R, then S and T. What happens in case of translocation? A part of chromosome is broken. Say for example C, D, E, it will be broken. Okay, it will get broken and it will get joined in other non-homologous chromosome, ultimately leading to the formation of what? A new one, that is O, P, then Q, okay. After that C, D, E, then R, S, T. So like this. So here it will be what? Transpositioned or translocated, okay, complete change in the position or location. So that type of what aberrations can also be seen. So these all are termed as what? Different types of chromosomal aberrations, okay. All of these are termed as chromosomal aberrations. Is that clear? So next one, coming to the next part of the chapter, that is the point mutation. Okay, point mutation. What is point mutation? It is a type of mutation that occurs at only one particular point on the DNA. Okay, at only one point on the DNA. Point mutation. What is point mutation? Okay, it is a type of mutation which occurs only in one point of the DNA. Okay, only in one point means only with respect to one nitrogenous base pair. Okay. If it occurs with respect to only one nitrogenous base pair, then it is called as point mutation. Okay, it is called as what? Point mutation. Okay. See here, these are the changes that occur in an individual gene, and these are due to structural change in the DNA molecule at single locus. Very important point to be noted. Okay. They are occurring due to what? Change in what? The single locus, and these alter the message conveyed by the genes, because they will lead to change in what? A particular amino acid, okay, replacement of a particular amino acid. When a particular amino acid is changed, it will lead to the defect in that protein which will be produced. Clear? And a single nucleotide will be altered here, right? Next one, the point mutation is of two types, that is substitution mutation and frame shift mutation. Point mutation have got two types. One is substitution, another one is frame shift. We have already discussed about the substitution mutation and frame shift mutation in the different chapters, in the earlier chapters, or else we will discuss the same thing again in the next chapter, that is molecular basis of inheritance. Okay. In both the chapters you are going to study about what? The mutations. And let us start with this one. What is substitution mutation? By the name itself you can understand what do you mean by the substitution type of mutation. Here substitution will take place. Okay. And what is that substitution? See, replacement. Substitution is nothing but what? Replacement. And in the substitution mutation, what happens? Replacement of one nitrogenous base by another nitrogenous base will take place. Okay. Replacement of one nitrogenous base with another. Okay. One nitrogenous base will be replaced by another, that is called as substitution. And in this there are two types. Okay, in this there are two types which are not mentioned here. Okay, in this there are two types where adenosine and guanosine, it means these are purines, and the cytosine and thymine, these are pyrimidines. What may happen in the substitution? A purine may be replaced by another purine, or purine may be replaced by the pyrimidine. If a purine is replaced by another purine itself, okay, then it is called transition. Okay. And if a purine is replaced by another one, translocation it may be called as, right. So different types of what? The mutations are there. In detail we shall discuss in the next chapter. Okay. Next one, frame shift mutation here. What is frame shift mutation? Here also the changes are occurring at only one particular nitrogenous base, but the change which is occurring may be what? Addition or deletion. Okay. by means of addition of a nitrogenous base or by means of deletion of a nitrogenous base, what will happen? The frame of the complete DNA, okay, will be shifted to its right side or else will be shifted to its left side. Due to that, it may lead to change in the complete sequence of the what? Amino acids. Okay, due to that, it may lead to change in the complete sequence, okay, complete change in the arrangement or complete change in the reading of the genetic code. So that is called as what? Frame shift mutation. What is frame shift mutation? It is a type of point mutation, okay, in which, okay, it is a type of point mutation in which the complete what? Sequence of the amino acids which will be produced, okay, the complete sequence of the amino acids or complete sequence of the nitrogenous base pairs of that particular gene will be altered, or the complete nitrogenous base sequence will be altered. Okay. Why the complete nitrogenous base sequence will get altered? Because by means of addition or by means of deletion of one or more nitrogenous bases, okay, one or more nitrogenous bases, what will happen? The shift of the frame will take place. Say for example, I will take an example here, okay. See, Ram has the same NCERT example I am taking. Ram has red cap. Ram has red cap. It means this particular sentence can give a particular meaning. Okay, a particular sentence will give a meaning. If anything is to be deleted, and this sentence I have written here based upon the genetic code. Okay. In the next chapter you will study about genetic code. So let us not take the example. I will take a common example here. So if you write the sequence of the nitrogenous bases, okay, say for example on the DNA, if you consider the nitrogenous base sequence, okay, that is A, T, C, G, then C, okay, C, A, C, A, T, T, A, T, A, G, G, like this, okay. This is what the nitrogenous base sequence of one particular DNA strand, on one strand, okay. On one particular DNA strand, if it is the nitrogenous base sequence, then by means of addition or by means of deletion, the frame will shift to its left side or else the frame will shift to its right side. See here, in this particular sequence of the nitrogenous base pair, if I delete the G, okay, if I delete the G, then what will happen? All the further nitrogenous bases which are present, they will shift one step back. Yes or no? They will shift one step back. That is called as frame shift mutation. The frame of the complete set of this particular DNA fragment will be shifted to its back, right? That is frame shift. And at the same time, if any addition takes place, okay, here the G, if it is deleted, then it will shift to the left side. If any addition takes place, say for example, if I add another C here, if I add another C, then what will happen? The complete frame will shift to the next part. Is that clear? Complete frame will be shifted to the next one, okay, or its right side. So that is called as what? The frame shift mutation. And what happens if by addition or by deletion, when the frame shifts to its right side or left side, what will happen? See, when the frame is shifted to its left side by means of deleting the G, then what will happen? The amino acids can be changed. How? Because this particular change or this particular deletion will lead to shifting of this frame, so that the nitrogenous base sequences on the mRNA, that means the codons which will be produced, they will be altered, okay, the codon will be always in triplet form. That is why we could have read GCA if it is not deleted, but when it is deleted, the codon can be read as CAC. We will shift to the next one by deleting this one. That is why the complete further sequence will be changed. Okay. So, that will lead to what? The particular change in that point mutation, okay, frame shift mutation. Clear? Next one is pedigree analysis. Then what is pedigree analysis? See, study of, okay, pedigree analysis, the study of ancestral history of a man for what? Transmission of genetic characters from one generation to the next is called as pedigree analysis. Okay, very important point to be noted. What is pedigree analysis? Study of ancestral history of man, of transmission of what? Genetic characters. That means a particular character, how it is transmitted in a particular family. To study that, we will plan a particular what? A planning, or we will put a graph, and that particular graphical representation or schematic representation of transfer of trait in a given family is called pedigree analysis. And for this pedigree analysis, these are the particular symbols that you have to remember, where square represents normal male and a circle represents normal female, and a connecting line between square and round or else circle will represent the mating, and this particular branches will represent the children, okay, and the children, if it is square represented by a square, it will be a male children or male child. If it is represented by a sphere, then it is a female child or girl, right, and these are dizygotic twins. Okay, dizygotic twins, and here monozygotic twins, where you can find one line which will be attaching them. Okay, and if you find the rhombus, it will be what? Sex unspecified, right? Sex unspecified. So, like this, and if it is written here, it is called as death. Then in a circle, if you find a dot, it is carrier. Then in this, if you find half shaded on one particular left side, then it is heterozygous, and if it is completely shaded, it is affected individuals, right. Like this, even in the NCERT it is given, you can observe, okay, you can observe in this particular video, okay. And further about this we shall continue in our next video, okay, we shall continue further in our next video, right. Thank you.